I am Dr. R. Krishnan, an assistant professor, PG and research department of microbiology, St. Joseph College of Arts and Science, Autonomous Cuddalore. Today I am going to present a topic on Salmonella typhi. Let's start with some introduction about Salmonella. Salmonella is one of the major foodborne pathogen that cause systemic or enteric infection of affecting approximately 2 million people worldwide each year. The first organism, Salmonella coalariasis, was isolated from a pig intestine by a scientist named Dr. Daniel Solomon. Salmonella belongs to the member of the family called Enterobacteriaceae, and only two types of or two species of Salmonella, namely Salmonella enterica and Salmonella bungori, are pathogenic and cause illness to humans. The disease which is caused by salmonella is called salmonellosis. Salmonellosis is a series of diseases that has a clinical spectrum of gastroenteritis and typhoid fever. Salmonella typhi and Salmonella paratyphi A is responsible for causing a typhoid fever and usually common in South and Southeast Asia. Salmonella typhimurium and Salmonella enteritidis or non-typhoidal salmonella and cause invasive bacterial diseases mostly common in sub-Saharan Africa. The fatality rate of salmonellosis is less than 1%, but malnourished children, immunocompromised patients, adults with HIV or at high risk. So next is some of the characteristics of Salmonella. So it is a gram-negative bacteria. It's a facultative anaerobes, motile, rod-shaped bacteria, a non-spore former. It's a non-capsulated except Salmonella typhi and Salmonella paratyphi. The growth temperature ranges from 5 to 45 degrees Celsius, but the optimum will be 35 to 37 degrees Celsius. They have a broad range of pH 3.8 to 9.5. They are resistant to bile salts and produce H2S. So next is the sources of transmission of salmonella diseases. Poultry is a major reservoir of salmonella and is transmitted to other organisms through fecal oral route. The salmonella species are also present in gastrointestinal tract of many animal, birds and reptiles. Salmonella was first isolated from the intestine of turkey flocks and chicken broiler flocks. The water and foods get contaminated from the feces and or urine of the infected human or animals. Consuming contaminated water and food infects the healthy individual and cause infection. The person to person also get infected through fecal oral transmission and or by handling contaminated food products or utensils and poor hygiene practices. Consuming tropical fish from contaminated water, raw meats, eggs, milk and milk products, salad dressings, cake desserts and topping, cocoa, peanut butter and chocolates are the possible source of salmonella infection. So next is the pathogenesis of salmonellosis. So for an organism cell to cause infection, 10 to the power of 6 to 10 to the power of 9 of viable cells must be ingested. After the ingestion of bacterial cell, the organism must survive a host defensive barrier such as salivary bactericidal enzymes, the hydrochloric acid of the stomach, lysozyme, bile salts, intestinal proteases, and gut microflora. The use of oral antacids and antibiotic decreases the gut microbiome, which facilitates the growth of salmonella. Many factors are determined for the salmonella to invade inside the epithelial cells that include the type 3 secretion system, fimbriae, flagellin, and bacterial DNA. The pathogen attaches to the epithelial cell within Peyer's patches with the help of fimbriae. Then endocytosis occurs where the pathogen gets engulfed by the cells and passes through epithelial cells within a membrane bound vacuole. They multiply in the vacuoles and releases into the mucous membrane and the inflammatory cells and toxins are produced, which releases prostaglandins. The enterotoxin produced by Salmonella activates the adenylate cyclase that stimulates intestinal secretion. In systemic enteric fever, the macrophages take up the invaded bacteria to the mesenteric lymph nodes where the organism multiplies and enters the bloodstream causing bacteremia. So next is the lab diagnosis of salmonellosis. So there are two or three different methods will be used to identify this bacteria from the infected person. First is called as a culture method. The commonly used lab media for the isolation of salmonella species are deoxycholate citrate agar or XLD agar or SS agar and McConkey agar. If the sample is minimum, enrichment media are used such as tetrathionate or selenite F broth will be used. So next is serological test. So viral agglutination test is done which measures the antibodies against the antigen of the pathogen. It is determined against the somatic O and flagellar H antigen of the organism. So finally the treatment for salmonellosis. So antibiotics are not recommended for Salmonella enterocolitis, but in case of enteric fever and typhoid fever, antibiotics such as ciprofloxacin, azithromycin, ceftriaxone can be used. Thank you.